Hello, today's class you'll be learning about drug metabolizing enzymes. So before this, we'll just see what is metabolism. Okay, metabolism is, in another words, it is also called as biotransformation. Okay, metabolism means biotransformation, that is the alteration the alteration of the drug inside the body. So this is basically metabolism in that sense. If you see, this biotransformation is a process where there will be some chemical reaction in the drug, so that this drug is converted into more polar water soluble compound, so that it can be easily excreted from the body. So this is also the definition of biotransformation, very important. And where does this occur? It mainly occurs in the liver. This metabolism of drugs mainly occurs in the liver. Apart from liver, kidney, intestinal mucosa, lungs, blood and skin. To minor extent, these are the other organs of metabolism. And here I have also mentioned few examples. Some drug which is metabolized in the plasma, that is succinylmethonium, okay, which is metabolized by plasma choline esterases. This enzyme, it is metabolized. And in the gut, tyramine and salbutamol get metabolized. Also in the lungs, you have prostanoids which are metabolized. So these are few examples. So moving on to learning in detail about what is biotransformation. So biotransformation, as we also saw, it was basically metabolism, right? So what happens in the metabolism is something, there will be formation of metabolites. So in the process of this, an active drug, which was an active drug which you have taken, can be converted into inactive form. Not only this, an inactive drug, that is the prodrug which you consume, can be converted into its active metabolite. After the action of this metabolite, this can be again deactivated. Okay. Also, an active drug can be metabolized to and more active drug can be metabolized to another active metabolite, which is also having the same effect or more effect than the prior drug. Okay, and then it can be inactivated finally to form a polar soluble compound, so that it can be easily excreted. So all these process occurs during the process of biotransformation. Here are few examples for different things what is happening in biotransformation. The first thing what we saw was an active drug which is converted into inactive form, like morphine, which is an active drug. It will be converted into inactive metabolite which is easily excreted. Also chloramphenicol, these also will be inactivated after biotransformation. Similarly, an active drug to active metabolite. So digoxin is also an active metabolite, and digitoxin is also an active metabolite. Digitoxin is converted to digoxin. Okay. And diazepam, oxazepam. Diazepam, after its metabolism or biotransformation, is converted to oxazepam. Even oxazepam also has some anti-epileptic or activity. Okay. So these two or an example of conversion of active drug to an active metabolite. Also, an inactive drug which is a prodrug will be converted into active metabolite. This inactive drug or prodrug does not have any action. Only after its conversion into its active metabolite it will produce the desired pharmacological action. Very important example is levodopa, which is converted into dopamine. This is given in case of Parkinson to overcome the loss of dopamine in the periphery by preventing its degradation by decarboxylase enzyme. Whereas levodopa is not degraded by dopamine decarboxylase, so that more the desired amount of dopamine can be available at the basal ganglion for the treatment of Parkinson's. So that is the importance. Similar way, prednisone is converted into prednisolone, that is the active compound. Enalapril, which is an prodrug, is converted into its active form, that is enalaprilant. Biotransformation, as I was telling, there will be metabolism and all that, and this metabolism is mainly catalyzed by few enzymes which are involved in biotransformation, and they are specific enzymes. Okay, and these enzymes are divided into microsomal enzymes, okay, and non-microsomal enzymes. Okay, and this microsomal enzymes are mainly present in the liver microsome. Apart from this, to small extent in the cytoplasm and mitochondria of liver also there is enzymes in liver microsomal enzymes. Non-microsomal, as we have already seen, it is seen in the plasma and other areas. So this biotransformation reactions are classified into two phases of reaction, phase 1 and phase 2. Phase 1 reaction is phase 1 reaction is also called as non-synthetic reaction. Either way they can ask. And phase 2 reaction is synthetic reaction. What is the synthetic? Give an example of synthetic reaction, like that they can ask. Okay, so this is what happens. So whenever you consume a drug, so it can undergo phase 1 or phase 2, or it can be excreted unchanged. Okay, phase 1 metabolites usually are reactive, and then they get conjugated through phase 2 and then excreted. We'll also see this in the next slides. So this is the biotransformation reactions. These are the reactions. Phase 1, oxidation, hydroxylation, dealkylization, deamination, hydrolysis. These are phase 1 reactions. Phase 2, conjugation reactions. Various conjugations reactions are there. We will see. So as I mentioned earlier, after a drug is metabolized through phase 1, usually highly reactive substance will be produced, and this get conjugated with the endogenous substrate of which is obtained from carbohydrate or amino acid into a more polar ionized compound which can be easily excreted through phase 2 reaction. So moving into detail about different phase 1 reaction, to begin with the oxidation reaction. As the name suggests, there will be oxidation, that is addition of oxygen or negatively charged compound, or you can remove a positively charged ion by removal of a hydrogen compound. Okay, various drugs which are undergoing oxidation process, it is mentioned here. Some are containing some get oxidized at the carbon atom, or some get oxidized at the nitrogen, phosphorus or sulfur atom. So based on this you have examples. Carbon atom oxygenation is brought about in drugs like barbiturates, phenothiazines, imipramine, okay, paracetamol. All these are various other examples, and this is catalyzed by the microsomal enzyme, that is CYP450 enzyme. Okay, this is also very important. And NADPH, that is dehydrogenase enzyme, CYP450 reductase, okay, molecular oxygen, okay, all these are very important enzyme names. Monooxygenase enzyme, and cytoplasmic and mitochondrial enzymes. Very important these names are. So they can give some other name along with this and ask you which is not the enzyme. Okay, so all these enzymes you have to remember in such cases. And condition where there will be oxygenation at the nitrogen, phosphorus or sulfur containing group atom, it is in case of ranitidine, cimetidine, clonazepam, adrenaline, alcohol. All these also get oxidized through oxidation process. Next, phase 1 reaction is reduction process, which is opposite to oxygenation. That is, there will be removal of oxygen atom, or and this process occurs in case of chloral hydrate, chloramphenicol, halothane and warfarin group of drugs. Hydrolysis, next phase 1 reaction. This as the name suggests, there will be addition of an water molecule, and the drug get metabolized with the enzymes, that is esterase, amidases or peptidases. Okay, these enzymes. And this kind of reaction occurs with procaine, lidocaine group of anesthetics, local anesthetics, aspirin, carbamazepine. Okay, all these group of drugs will undergo hydrolysis. Cyclization and decyclization reactions are also phase 1 reactions. Cyclization is a process where a ring is formed from a straight carbon atom. Okay, and this occurs with proguanil, which is a anti-malarial drug. Decyclization is a process where a cyclic ring-like structure is opened up. Okay, opening of ring of cycle molecule. So this occurs in case of barbiturates and phenytoin. Alteration in this chemical structure also deactivates the drug. Moving on to phase 2 reaction, very important phase 2 reaction is glucuronide conjugation. So there will be n number of questions. Okay, this is the only phase 2 reaction which is present or which will occur in the microsomal enzymes. Okay, so that is why this is very important, and this reaction will occur with drugs containing hydroxyl or carboxylic acid group. What happens with this group of drugs? So under the influence of UDP glucuronide transferase enzyme, so the drug will combine with the glucuronide to form drug glucuronide complex, which is easily excreted from the body. So examples are chlorpheniramine, aspirin, okay, paracetamol, lorazepam, morphine and metronidazole. Apart from this, endogenous substance which is also which will also undergo glucuronide conjugation is bilirubin, and hormones like steroid and thyroxine also undergo glucuronide conjugation. Moving on to the next phase 2 reactions, we saw glucuronide conjugation now, and then we are moving on to acetylation. Okay, acetylation process where this usually occurs with compounds which contain amino or hydrolysine residues. Okay, so compounds which contain amine or hydrolysine residues, they get conjugated with acetylcholine and undergo acetylation process. Examples include sulfonamides, isoniazide, PAS, hydrolysine, clonazepam, all these group of drugs. Next phase 2 reaction is methylation process where methionine or cysteine is added to get and the drugs containing amine or phenol groups get methylated. Examples are adrenaline, histamine, nicotine, methyl dopa, all these group of drugs. And phase 2 reaction, next we also have sulfate conjugation which occurs with phenolic or steroid compounds. Examples are chloramphenicol, methyl dopa, adrenaline and sex hormones. Glycine conjugation, these are minor pathways. This mainly occurs with salicylate and other drugs which contain carboxylic acid. Okay, and they get conjugated with glycine. Few other minor pathways include glutathione conjugation, important. This mainly occurs with paracetamol, very important question, that is paracetamol undergoes glutathione conjugation. So whenever this glutathione levels falls, there will be toxicity which will result in tissue damage, but this is a minor pathway but an important type of conjugation which is important in paracetamol metabolism. Also ribonucleoside and nucleotide synthesis, these are mainly important in the activation of purine and pyrimidine antimetabolites. Okay, these usually are prodrugs which are later converted into active form, purine and pyrimidine containing antimetabolites which are used in the treatment of cancer. So they also get they undergo phase 2 reaction and they get activated. So with this we have covered phase 1 and phase 2 reaction. So the next important thing which we are seeing here is something called as Hoffman's elimination. So Hoffman's elimination is a unique type of metabolism of atracurium. Okay, what happens here is there will be the drug, this atracurium, get inactivated in the body fluids by spontaneous molecular rearrangement. Okay, there will be spontaneous molecular rearrangement of this drug when it comes in contact with body fluids without the use of any enzyme. Okay, so non-enzymatic degradation example is atracurium, which will use this Hoffman's elimination. Okay, so this is very important question. Usually they keep on asking this atracurium. So we were reading about microsomal enzymes and all that. We saw what it was doing. So we'll see in detail, little detail about these things, because it is also a question. So these microsomal enzymes, where are they present? We saw it was in the smooth endoplasmic reticulum. Okay, it is mainly present in the smooth endoplasmic reticulum of mainly liver. Okay, in the liver, smooth endoplasmic reticulum, you see all these microsomal enzymes, and they catalyze most of the phase 1 reactions like oxidation, reduction, hydrolysis, and only one phase 2 reaction, that is glucuronide conjugation.